Good morning. In this video, I am going to discuss about tryptophan metabolism. This tryptophan, in fact, actually any amino acid metabolism, I will be discussing under the following headings. To start with, a brief introduction about the amino acid. Next, how they are synthesized. After synthesis, how they are catabolized in our body. Between this anabolism and catabolism, what are the functions it executes? If these functions are not done, what are the disorders it may produce? So these are the headings under which I will be discussing about tryptophan metabolism. First, let me start with a brief introduction about tryptophan. Amino acids are generally classified based on four different categories. The first category, the first classification is based on structure. So when you see the structure of tryptophan, it is an indole group made up containing, it is an amino acid which contains indole group as a special group, which is nothing but an aromatic ring. So this amino acid is called as aromatic amino acid. So this aromatic tryptophan, when you see, its structure is almost similar to phenylalanine. In phenylalanine, there is a benzene ring or a phenyl ring with which alanine is attached, but whereas here it is an indole group to which alanine is attached. Okay, next, the second classification is based on the solubility of the side chain. Here the side chain or the indole group doesn't have any polar group. So we call this tryptophan as a non-polar or hydrophobic amino acid. Next, the third classification is based on the synthesis. Based on synthesis, amino acids can be broadly classified into essential and non-essential amino acids. Essential means it can be synthesized in our body and no need to supplement, and it can, sorry, essential means it cannot be synthesized in our body and it has to be supplemented only through diet. Whereas non-essential means it can be synthesized in our body, so it is not essential in the diet. Okay, so there are various mnemonics to remember these essential and non-essential amino acids. Those things we have already discussed in the previous videos. So from that you can easily make out that tryptophan is an essential amino acid. That means it cannot be synthesized in our body and it has to be supplemented only through diet. Okay, next, the last classification is based on the catabolic fate. After catabolism, the tryptophan can be broken down into, they can be converted into the end products of tryptophan catabolism can be utilized for both glucose synthesis and also for ketone body synthesis. That is why it is called as both glucogenic as well as ketogenic amino acid. So these classifications you should know. Okay, so our tryptophan belongs to which type of amino acids? Okay, under these four categories, you should be knowing it. Next, apart from producing glucose and ketone bodies during their catabolism, the tryptophan can also be used for the synthesis of various biologically important compounds like serotonin, melatonin, and vitamin niacin. Okay, so how all these are synthesized and how the tryptophan is catabolized in our body, we will discuss in detail in the coming slides. Okay, so this is about the introduction. Next, coming to the second heading, anabolism. That means synthesis. Just now we discussed that tryptophan is an essential amino acid, which means it cannot be synthesized in our body. So I am skipping anabolism and directly going on to catabolism. Okay, so based on catabolism, we saw that tryptophan is a both glucogenic as well as ketogenic in nature. That means the end products can be utilized both for glucose synthesis and also for ketone body synthesis. Okay, so how it happens we will discuss in detail now. The two major enzymes which play key role in tryptophan catabolism are, one is tryptophan pyrrolase, the enzyme tryptophan pyrrolase, and the other one is kynurenase. These are the two enzymes which play a major role in tryptophan catabolism. In this, tryptophan pyrrolase, which is an iron containing enzyme, what they will do means they will they will act on the indole ring and they will open the ring. The small, if when you see the indole ring, there are two rings are there. One is a typical benzene ring and the other one is a five-membered ring. So this five-membered ring will be oxidized and will be broken to form a compound called as N-formyl kynurenate. Okay, so this is the first step in tryptophan catabolism. An iron containing tryptophan pyrrolase enzyme will act on tryptophan and converts it into N-formyl kynurenate. Next, the second enzyme which plays a key role in tryptophan catabolism is kynurenase. But this kynurenase will act only on hydroxykynurenine, not on a formylkynurenine. So what we have to do, the formal group has to be replaced by hydroxyl group. So how we can do that means, in the first step, the formal group will be removed by tetrahydrofolate acid. Okay, the enzyme formylase will remove the formal group from kynurenine and it will be released as formal tetrahydrofolate, so that N-formylkynurenine will be converted to kynurenine. Next, in the subsequent step, by an enzyme called as hydroxylase, and hydroxyl group will be added to kynurenine to form 3-hydroxykynurenine. So once this 3-hydroxykynurenine is formed, the enzyme kynurenase can act on it. Okay, so when it acts on it, it will break the 3-hydroxykynurenine into two compounds. One is 3-hydroxyanthanolic acid and the other one is alanine. Okay, in this, the 3-hydroxyanthanolic acid form, it can be converted to acetoacetyl-CoA and it can be utilized for ketone body synthesis. Whereas the alanine form, it can by transamination reaction, it can be converted to pyruvate and that pyruvate in turn can be utilized for gluconeogenesis. That is the reason we call tryptophan as both glucogenic as well as ketogenic in nature. Okay, so this is how tryptophan is catabolized in our body. So here I would like to give you a small clinical significance. I said there are two enzymes which play a key role in tryptophan catabolism. One is tryptophan pyrrolase, which is an iron containing enzyme, whereas the other one is kynurenase, which needs vitamin B6 or PLP as its coenzyme. So what will happen in B6, vitamin B6 deficiency means this kynurenase will not work. So the kynurenine will be accumulating in our body. So this accumulating kynurenine will go for an alternate pathway and it will be converted into xanthurenic acid. So in vitamin B6 deficiency, there will be excess production of xanthurenic acid which will be excreted in the urine. So in vitamin B6 deficiency, this can be used as a marker. So urinary xanthurenic acid levels will be elevated in vitamin B6 deficiency due to the defect in the tryptophan catabolism. That can be used as a marker to detect vitamin B6 deficiency. Okay, so that's it about the catabolism of tryptophan. Apart from this, there is also one more minor pathway for tryptophan catabolism which actually takes place within the intestinal cells. Okay, it will be taking place in the intestine. Okay, so here actually normally what will happen, the tryptophan is an essential amino acid, so it has to be supplemented in the diet. So when you supplement it through diet, this tryptophan will be absorbed in the intestine. So if it is observed, it will go for the major catabolic pathway. Suppose if it is not observed, it will be oxidized or catabolized within the intestinal cell by this minor pathway. So in this minor pathway, what is the main enzyme or the first enzyme to act means tryptophan transaminase. Okay, transaminase, we all know that it is an important enzyme which plays a key role in the catabolism of any amino acid. Okay, so what is the action of the transaminase means? It will remove an amino group from an amino acid and it will convert it into its corresponding keto acid. We all know that tryptophan is nothing but a indole group with alanine. Okay, from that alanine, if you remove the amino group, it will be converted to its corresponding keto acid called as pyruvate. Okay, so tryptophan, upon transamination by tryptophan transaminase, they will be forming indole pyruvic acid. Now, this pyruvic acid is a 3-carbon acid, is a 3-carbon keto acid. Okay, so these 3 carbons will be removed one by one in the subsequent steps. First, the first carbon will be released to form the indole pyruvate. The pyruvic acid, 3-carbon pyruvate, will be converted to indole acetic acid. Next, again one more carbon atom will be removed from this acetic acid so that it will be converted to scatol. Okay, and this scatol forms the major excretory form in the feces. Okay, so the tryptophan which is not absorbed by the intestine will be converted to scatol and it will be excreted through feces. Sometimes instead of getting excreted in feces, this scatol may be absorbed by the intestine. It can enter into the body. Okay, so after it enters into the body, the one more carbon atom of the pyruvate will be removed from it and the scatol will be converted to indole. This indole in turn will be detoxified in the liver. Okay, so detoxification means phase 1 and phase 2 reaction. Phase 1 is oxidation, it will be oxidized, and in phase 2 reaction it will be conjugated with a sulfur atom and it will be converted into indicon, which is nothing but the potassium salts of indoxyl sulfate. Okay, so this indicon, after detoxification, this indicon will be removed from our body through urine. Okay, so this is what happens in the minor metabolic pathway. Okay, so what is the difference between major and minor catabolic pathway means? So when the tryptophan is absorbed, normally it will go for the major catabolic pathway. So what is the end product in the major catabolic pathway? It will be diverted for glucose as well as ketone body synthesis. Whereas if it is not absorbed by the intestine, what is its fate? It will be converted to scatol and excreted in stools, or otherwise it will be converted to indicon and it will be excreted in the urine. Okay, so these are the various end fates of tryptophan in our body. Got it? Yes. Next, after catabolism, next we will see what are the other functions before being catabolized by our body. What are the functions it executes in our body? So this tryptophan is needed for the synthesis of three biologically important compounds. So what are those three biologically important compounds means? To start with, it is serotonin. Okay, so it is actually the three compounds are serotonin, melatonin and niacin, vitamin niacin. Okay, so these are the three biologically important compounds produced from tryptophan. So now here we will see how all these compounds are synthesized from tryptophan and what are their biological functions. That is that you should be knowing about each one of this compound. So first we will discuss about serotonin. Okay, so what is the other name of serotonin means? It is nothing but 5-hydroxytryptamine. This is the other name for serotonin. Now we will see how the tryptophan can be converted to 5-hydroxytryptamine. Okay, so as the name says, first we should add an hydroxyl group at the fifth position so to form 5-hydroxytryptophan. This will be done by an enzyme called as tryptophan hydroxylase. So this tryptophan hydroxylase is exactly similar to that of phenylalanine hydroxylase. That is why it uses the coenzyme tetrahydrobiopterin. Okay, so this enzyme along with its coenzyme will add an hydroxyl group at the fifth carbon atom and convert tryptophan to 5-hydroxytryptophan. Okay, next in the subsequent step, this 5-hydroxytryptophan will be converted to 5-hydroxytryptamine. Okay, so that means an amino acid tryptophan should be converted to its amine tryptamine. Okay, so how it can be done? Amino acid to amine. So what you should do? You should remove the acid group. Okay, that means the decarboxylation should happen. So here this decarboxylation is done by an enzyme called as decarboxylase, which needs PLP or vitamin B6 as a coenzyme. Okay, so when this decarboxylase acts on 5-hydroxytryptophan, the acid group will be removed and it will be converted to its corresponding amine, which is called as 5-hydroxytryptamine or otherwise called as serotonin. So this serotonin formed in turn will be catabolized by monoamine oxidase. This amine or most of the amines in our body, it will be further detoxified or oxidized by monoamine oxidase. Okay, so this monoamine oxidase, what they will do, they will go for oxidative deamination and they will be removing the amino group from the serotonin and it will be forming 5-hydroxyindole acetic acid. So this is the product you will get from serotonin catabolism. Okay, so normally we saw various fates of tryptophan. Okay, normally it can be used for glucose synthesis, it can be used for ketone body synthesis. In the intestine, it can be converted to scatol or indicon. But along with that, sometimes the tryptophan can be converted to serotonin, and that serotonin in turn can also be converted to 5-hydroxyindole acetic acid. So this is also one of the end product we get from tryptophan metabolism. Okay, so that's it about that is it about the fate of serotonin, which is formed from tryptophan. But before getting metabolized or catabolized, they perform lot of functions in our body. This serotonin, it performs lot of functions in our body. Okay, so what are those functions means? The serotonin is mainly produced in 4 major tissues. One is brain cells, second GIT, third platelets, and fourth one smooth muscle cells. Okay, in all these tissues, the serotonin produced will go and bind to the serotonin receptors, 5-hydroxytryptamine receptors, and they will be executing various biological actions. So what are those actions means? To start with, in brain cells, our serotonin, it acts as a mood stabilizer. Okay, so it is the one which makes you happy and it prevents you from getting depression. Okay, so that's the reason for treatment of depression, this serotonin metabolism is highly targeted. The commonest drug used for antidepressants are MAO inhibitors, monoamine oxidase inhibitors. Okay, so what they will do, they will inhibit this monoamine oxidase, which is actually responsible for catabolism of serotonin. So if this enzyme is inhibited, the life cycle, half lifespan of serotonin will be extended, so that their action will be extended and it can act as an antidepressant. Second, the other two drugs, selective serotonin reuptake inhibitors and tricyclic antidepressants. Both these drugs, what they will do means they will increase the half-life of serotonin within the synaptic vesicles so that their action will be prolonged and it helps to prevent the depression. Okay, so thus serotonin plays an important role in stabilizing mood and preventing depression. Okay, apart from this, it also has few other functions in the CNS. One is it helps in regulating the sleep, appetite and temperature regulation also. In addition to that, it also influences memory and learning. So these are the various functions of serotonin as a neurotransmitter in the brain. Apart from the brain, it is also produced in few other tissues also. The second thing is GIT. So in GIT, what is the action of serotonin means? It will increase the intestinal motility. Whenever you consume some toxic chemicals or noxious substances in your diet, what will happen means that those substances should not be absorbed by the body. So what will happen means the intestinal mucosa cells, they will produce more and more of serotonin. So this serotonin in turn will increase the GI motility and they will flush away the toxic or noxious chemicals. Okay, so thus it helps. This is the physiological action of serotonin within the GI system. Next, coming apart from that, it is also produced in platelets, which will be used for coagulation and also for wound healing process. Then finally, in the smooth muscle cells, it helps in smooth muscle contraction. That is the reason it is considered as a potent vasoconstrictor. Okay, so that's it about the synthesis and functions of serotonin. Next, we will move on to the next biologically important compound, melatonin. Okay, so how this melatonin is synthesized means it is synthesized from serotonin. First, from tryptophan, serotonin will be formed, and from that serotonin, by two steps, it can be easily converted to melatonin. So how it will be converted means in the first step, an acetyl group will be added to serotonin by an enzyme called as acetylase to form acetyl serotonin. In the subsequent step, a methyl group will be transferred to this by methyl transferase to form our melatonin. Okay, so this is how melatonin is synthesized from tryptophan. So first tryptophan should be converted to serotonin, and that serotonin, by adding an acetyl group and a methyl group, it will be converted to melatonin. Okay, so for these reactions, the donor for acetyl group is acetyl-CoA, whereas the donor for methyl group is S-adenosyl methionine, the activated methionine form. The S-adenosyl methionine acts as a donor for methyl group in the melatonin synthesis. Okay, so this synthesis of melatonin, it mainly takes place in the pineal gland and it helps in maintaining the sleep-wake cycle. Melatonin is actually the one which makes you to sleep nicely. Okay, so it will be produced during the night time, which makes you to sleep. But during the daytime, when the light is there, that can destroy the melatonin. The blue light, especially the light of blue light wavelength, can destroy the melatonin and it will make you awake. Okay, it will decrease the sleep and make you awake. Okay, so thus melatonin plays an important role in maintaining the sleep-wake cycle, or otherwise we can call it circadian rhythm. Okay, so this is one function of melatonin. In addition to this, recent studies have shown that melatonin also has some antioxidant property. Okay, so that's it about melatonin. Melatonin over. Next, the last product which we get from tryptophan is niacin, vitamin niacin. Okay, so how this is synthesized means during the catabolism of tryptophan, we saw that tryptophan will be broken by the enzyme tryptophan pyrrolase and kynurenase to form 3-hydroxyanthranilic acid and alanine. Normally, what is the fate of this 3-hydroxyanthranilic acid means it will be diverted for ketone body synthesis. But sometimes this 3-hydroxyanthranilic acid can be converted to quinolinic acid. Okay, and to this quinolinic acid, if you add a phosphate group and a ribose group, it will be converted to niacin. Okay, so this is how niacin is synthesized from tryptophan. Okay, one of the key enzymes involved in this process is QPRTase, quinolinic acid phosphoribosyl transferase. That is one of the key enzymes which is needed for the synthesis of niacin from tryptophan. Okay, so normally from every 60 milligram of tryptophan, 1 milligram of niacin can be synthesized by this pathway. Okay, so next, coming to the functions of niacin. So there are various active forms of niacin, NAD, NADP, and the reduced forms are there. So both these oxidized and reduced forms of NAD and NADP, they play an important role in various oxidoreductive reactions in our human body. So all those things you might have already studied under the vitamins. So I'm not going into deep into that. Okay, so that's it about the biological functions of tryptophan. So it is needed for the synthesis of three biologically important compounds. One is serotonin, the other one is melatonin, and the third one is vitamin niacin. Okay, next functions over. Next, moving on to the last heading, the disorders related to the tryptophan metabolism pathway. So there are three disorders related to tryptophan metabolism pathway. The first one is pellagra, which happens due to niacin deficiency. So whenever there is a deficiency of tryptophan or defective absorption of tryptophan, or whenever there is a problem in the pathway which converts tryptophan to niacin, it can lead to symptoms of niacin like dermatitis, dementia, and diarrhea, and ultimately if it is severe it can also lead to death. Okay, so 3D can lead to 4th D also. Okay, so that is the reason for pellagra in tryptophan metabolism. Okay, in defective tryptophan metabolism. So for the conversion of tryptophan to niacin, we need vitamin B6, which acts as a coenzyme for kynurenine. So in vitamin B6 deficiency, the synthesis of niacin from tryptophan will be grossly affected, and that can lead to pellagra also. Okay, so that's it about pellagra. Next, coming to the second disorder, it is carcinoid tumors. So these carcinoid tumors are nothing but tumors of a specialized cell called as argentaphin cells, which are present in the small intestine and appendix. Okay, that is why these carcinoid tumors are also called as argentaphinoma tumors. Okay, so when these cells are becomes malignant, what will happen means they start producing more and more amount of serotonin. There will be excess production of serotonin. So serotonin is produced from tryptophan. So when all tryptophan are utilized for serotonin synthesis, it may not be available for the synthesis of niacin. So these patients, they also will present with pellagra. Okay, but here in this patient, though they present with niacin, so how we can diagnose them means characteristically, as serotonin is produced in excess, the catabolic end product from serotonin, that means 5-hydroxy indole acetic acid levels will be increased in urine. Okay, so normally if it will be usually less than 5 milligrams per day, but if it is more than 25 milligrams per day, you can easily make the diagnosis of carcinoid syndrome. Okay, so that's it about carcinoid tumors or carcinoid syndrome. Okay, so here the tumor cells will produce more serotonin. So because of that, tryptophan deficiency will happen and pellagra will happen. Okay, so that's it about carcinoid tumor. Next, the third disorder is Hartnupp's disease. So in Hartnupp's disease, the transporter protein, which is responsible for the absorption of tryptophan in GIT and reabsorption of tryptophan in kidney, is defective here. So that is the problem in Hartnupp's disease. There is a defective transporter both in the GIT and also in the kidney. So since this transporter is defective, what will happen means the tryptophan cannot be absorbed by the dietary tryptophan cannot be absorbed into our body. Okay, so that means they will present mainly as tryptophan deficiencies, or tryptophan will not be there, so the products produced from them will not be there. So they will also mainly present with pellagra because niacin synthesis will be grossly affected. Okay, so this is regarding the defect in the intestinal transporter. On the other hand, so if there is a defect in the kidney transporter, what will happen? It may not be reabsorbed by the body. So tryptophan cannot be reabsorbed. So more and more tryptophan will be lost in the urine. In addition to that, there will also be excess excretion of urinary indole compounds, which can be detected by Obermeyer's test. So why there is excess excretion of indole compounds means, so in Hartnupp's disease, when the tryptophan absorption in GIT is not happening, what will happen? The tryptophan will accumulate in the intestinal cells. So this intestine, in the initial slides we saw that we have a minor metabolic pathway mainly by the action of bacteria. So this tryptophan will be converted to scatol and indole-like compounds, which will be excreted in feces and urine respectively. Okay, so that is the reason urinary indole compounds will be excreted in excess during Hartnupp's disease. Okay, so that's it about tryptophan metabolism. Okay, so to summarize, tryptophan is an essential amino acid which cannot be synthesized in our body, so it has to be supplemented only through diet. Coming to the catabolism part, it can be broken down by, there are two key enzymes in the pathway. One is tryptophan pyrrolase and the other one is kynurenase. So both, with the help of these two enzymes, tryptophan will be broken into two parts. One will be diverted for ketone body synthesis and the other will be diverted for glucose synthesis. Okay, apart from this ketone body and glucose synthesis, tryptophan can also be used for the synthesis of three biologically important compounds. One is serotonin, second melatonin, and the third one is niacin. Okay, so this synthesis and all these things may be affected in three important disorders. The first one is pellagra, second one is carcinoid syndrome, and third one is Hartnupp's disease. Okay, in all these three conditions, you will be having signs and symptoms of niacin deficiency or pellagra. Okay, why means tryptophan is essential for the synthesis of niacin. Okay, thank you. So that's it about tryptophan metabolism.